Good evening to all. I'm happy to spend time with all the participants here. This session is really meant for understanding that similar to HPLC, TLC also produces acceptable results in quantitative analysis. This I want to emphasize through this session. Second, both HPLC and TLC work under the same principle. There is no variation at all. Third is, TLC is comparatively very simple and versatile. And based on these three lines, today's session will be conducted. So please listen to me. Whenever you have a doubt, you please raise your hands. And as much as possible, I will immediately reply to all your queries. And if not possible, then we will take all those questions at the end towards the end of the session. With this explanation, I'm trying to take up my presentation. I'm planning to share the screen with you now. So let's get prepared for the presentation now. Topic is quantitative analysis by HPLC and TLC. Let me explain the basis of chromatography within 2-3 slides so that we will all think in the same language, because the terminology what I'm going to use should be familiar to you also. You might have understood the chromatography in a different terminology sometimes. So to make everybody to sit in the same platform, I'm going to explain what is a chromatography and the various terminologies being used in this line of subject. Okay, chromatography is a technique using which components of a mixer are separated, identified, and quantified. This chromatographic technique can be used for a purification of a compound from its impurities in production process, and in the laboratories, this technique is used for identification and quantification of various components present in a chemical mixer. We discuss analytical application of chromatography in this session. See, principles of chromatography. The chromatography works on several principles. Several principles are used in the chromatography, but we confine only with the two principles here. One is adsorption. Second is partition coefficient. See, the adsorption, in briefly, if you want me to explain, adsorption is the tendency of a molecule to stick to the surface of a solid material. And every component will have a different degree of adsorption towards a particular solid. And similarly, there is another principle which is called partition coefficient. This is the tendency of a solute molecule to get dispersed into different immiscible solvents. So with these two things, I'm not going to explain everything, what is partition coefficient, what is adsorption, but just I give an idea what is adsorption and what is partition coefficient. Just keep it in mind. It will be useful when we go through the session. And apart from these two principles, there are several other principles used in the chromatography, but we are not going to consider them. Some of such principles are ion exchange, gel permission and all, which we are not going to discuss in this session. We are going to discuss only based on the adsorption and partition coefficient. So what is the configuration of a chromatography? We talk about chromatography everywhere. What is chromatography and what does it contain? How does it work? If you ask this question, I'll say the chromatography system is having two important components. One is stationary phase and another is mobile phase. Stationary phase is normally a solid or liquid, and the mobile phase is normally liquid or gas. This mobile phase carries the sample, the mixture of components, through the stationary phase, where the components of the mixer experience the adsorption or partition, due to the difference of adsorption and partition of the solutes which are passing through the stationary phase, they start moving in a different speeds, so that when mobile phase carries this mixture of components, because of the difference in the adsorptivity or the partition coefficient, they are separated one after the other on the stationary phase. Then we use a suitable deductive mechanism to find out what are the components present in it and how many of them are detectable and what is the quantity, concentration of those things, which we'll discuss in the coming slides. And every one of us would have heard, just not just like the chromatography, we will come across the word called thin layer chromatography, liquid chromatography, solid, the column chromatography, gas chromatography, paper chromatography, and all. So these are all, there are no hard and fast rules. How to name each chromatography system, that here we are. I like to explain thin layer chromatography. See, it is named with their own ease. See, commonly known chromatography is, one is thin layer chromatography. In thin layer chromatography, stationary phase is thin layer of silica gel, and mobile phase is organic solvent. In the paper chromatography, the stationary phase is paper, and the mobile phase is organic or aqueous solvents. Aqueous solvents means it may be water, it may be ammonia, it may be a triethylamine, something like that. And the gas chromatography is, so the stationary phase is solid or liquid, and the mobile phase is gas. In this gas chromatography, where the solid is used as a stationary phase, which is packed inside a column, which is on this column will be coiled and it would have been fitted into the system. That coil will have normally an inert material inside, which will act as a stationary phase, and it will be sometimes solid, sometimes it will be liquid loaded on a solid material. So if it is purely solid, it is called gas solid chromatography, GSC, and if it is if the inert material contains liquid also, then it's called gas liquid chromatography. I like to explain the liquid chromatography with the next slide. This liquid chromatography I was trying to explain, it is nothing but something like a burette. You assume something like a burette, where you will fill with the silica gel and silica gel or alumina or magnesia. See, whatever the stationary phase we like to use, we can use. And at the top of it, we introduce a sample, which will be a mixture of different components, and we will pour the mobile phase from the top. It will come. See, because of the difference in the absorptivity, each component will move at a different speed. Although the mobile phase is going at a particular flow rate, constant flow rate, because of the difference in the absorptivity of each of these components, they will go in a different speed. So they will get separated automatically, and the one which is having at least absorption will come first. The one which is having the medium absorption will come second. The one which is having the highest absorption will come last. This is colored. We are showing the colored component only for understanding purpose. If it is not colored, we have to use every 0.5 ml or 1 ml. We have to collect and get it tested for the presence of the components, and accordingly we have to do, which we don't want to complicate at this point of time. This is just to understand the principle. I have shown this. So this is nothing but liquid chromatography or column chromatography. In this, as I explained, it is having solid as a stationary phase, which may be silica gel or alumina or magnesia. And again, the mobile phase is organic or aqueous solvents. In this also, we can use triethylamine, ammonia, and the buffer solutions, something like that we can use as aqueous solution solvents, and organic solvents will also be used. Okay, if the same liquid chromatography, if it is automated using the pump and the electronic backup, then it is called high performance liquid chromatograph. So I'm going to explain how HPLC will be looking like. The different components of HPLC is like this. It will have a pump, high pressure precision pump, and this precision pump will take solvents from the reservoir, which is mobile phase, and this blue colored tube, you can just watch the cursor moving. This blue colored tube is a stainless tube which would have been packed with the stationary phase, mostly silica gel, like almost like this. Okay, and the solvent will be taken from the reservoir by the pump, and it will be pumped through the column. And this outlet of the column will go to a detector, and where it is get detected, and the data will be captured in the computer. And in this spot, sample will be introduced. Just watch the cursor. So when this sample keeps moving, the solvent keeps moving, mobile phase keeps moving, sample is introduced, and the sample is taken inside the column, where it gets separated like this. This is a blue, red, green, and they will come into this detector. Mostly it will be a UV detector, where it will be detected based on the UV absorption. And if you see this computer, which is shown here, the screen, there will be a baseline will be very straight, flat, as long as there is no no component is coming out. When some component is coming out, there will be a peak will be seen like this. So if there are multiple peaks, it means that so many components are there. And this is how HPLC operates. So I hope this is a very simple explanation. I hope everyone would have understood if they are not familiar with that. Yeah, typical liquid photograph of the typical HPLC is like this. This is one compartment is a pump, and another compartment is a detector. The top one is a reservoir. Okay, and the straight line which you can see here, the white color one is a column, and the adjacent one is integrated, or earlier days it used to be called as integrated. Nowadays it is replaced with a computer software. So the software is loaded onto the computer, and whatever the data collected from the detector will be detected by this computer, and it will give a chromatogram like this. And you can see the several peaks, and each peak is coming out of this detector, and which will get recorded in this integrator or computer. And we can see on the screen this kind of chromatogram. And normally the peak area we will calculate. This peak, each peak is considered as a triangle, and using the formula for measuring the area of the triangle of AB, the peak area will be calculated by the computer, and the peak area would have been given here in the bottom. And there are 6 peaks are seen, prominent peaks. All those peaks are corresponding to different gingerols. Okay, I'm skipping to the next slide. This is a thin layer chromatographic system. In the system, if you see, this is a developing chamber, which is in something like a stoppered beaker, where some mobile phase would have been already placed. It will be of the height maybe 2 mm or 3 mm. And this is a white portion is a plate, thin layer plate. A thin layer of silica gel is coated on aluminium, and over which the samples are applied as spots. And this lower portion of that plate is immersed in the mobile phase in the developing chamber, and we allow the mobile phase to rise through the capillary rise. And during such a rise, the components in the samples applied here get separated. And after separation, they will be looking like this. They will be separated and looking like this. Okay, so let us, for time being, let us avoid those things, and we'll go back to the regular analysis. Yes. Now, as I told you earlier, we are going to explain three segments. One is, first I'm going to explain analytical procedure which is semi-quantitative analysis by TLC. And the next portion will be, demonstration will be quantitative analysis by TLC. And third one is comparison of HPLC results with the TLC results. Okay, now I'm taking semi-quantitative analysis by TLC. So the semi-quantitative analysis by TLC was an official method in Indian Pharmacopoeia of 1985 version. I don't know whether still it is an official method or not, but it was really an official method in 1966 version as well as 1985 version. Under the topic of normally it is given as a procedure under related substances, which was a limit test. Actually, in this method, which is an official method of Indian pharmacopoeia, it is normally used to detect impurities, related impurities present in API or bulk drug, to understand if a particular impurity is present beyond a level or not. That is, beyond a level means they will specify the quantity of the impurity should not be more than 0.1% or 0.2% or 0.3%. So this method is designed in such a way that after the end of the analysis, we can say whether impurity is there or not. If it is there, that impurity is more than the specified limit or not quantitatively, but we will not specify exactly what is the percentage of impurity. We can say only whether it is within the permitted level or not. So that's the reason it is called semi-quantitative. And to explain, I'm going to explain this procedure here. For that purpose, I'm taking a case study, estimation of bis dimethoxy curcumin in a curcuminoid complex, which is an extract of turmeric. So before getting into that, I want to explain, or I want to say what is curcuminoid complex. When you extract turmeric, we will get the crystals, curcumin crystals, and when we see it's a pure crystal, but when we analyze, we can find that it is a mixture of 3 different components. It's not a single compound alone. It will have curcumin, which is having 2 methoxy groups, and it will be another component in that will be a dimethoxy curcumin. Okay, is it audible for everyone?
Yes, sir.
Yes,
you can go.
Yeah.
Go ahead.
Yes, sir.
And the third one is this in the curcumin complex. It's a mixture of 3 components. One is curcumin, which will have 2 methoxy groups, and another will be having only 1 methoxy group, and the third one will not have any methoxy groups. So hence it is named as a bis dimethoxy curcumin. See, normally the composition will be like this. The first one, pure curcumin, will be around 75 to 80%. The second one will be around approximately 15%, and the third one will be approximately 2 to 3% or 2 to 4%. So now we are taking a curcuminoid complex crystals and going to estimate what is the proportion of bis dimethoxy curcumin present in the curcumin complex. Okay, so for that purpose, we are taking, just to look at the tabular column S1, the solution ID S1, 0.1 gram of curcumin complex is dissolved in 10 ml of acetone. That becomes 1% solution. And this solution is further diluted to 0.01, 0.02, 0.03 and 0.04%. And they are named with a different ID, S1, S2, S3, S4, S5, S6. And the 10 microliter of each of the solutions except S2. S2 is not required for this analysis. It is only for preparing the serial dilution. So S2 we are not applying. So S1 to S6 without S2 are applied here. Each application is of 10 microliter very accurately using a micro syringe we have applied. Okay, after applying and before developing, the plate will be looking like this, which is seen on the right side of the screen. Just watch this series of photographs, which will take approximately 1.5 minutes. This will give an idea how TLC is carried out. This is the preparation of solutions. And the chemist is drawing sample using a micro syringe. And this is getting prepared to apply on the plate, first solution. And she is applying the first solution on the plate. And after applying series of solutions, this is the photograph of the last solution being applied. After application, before development, the plate will be looking like this. and she is pouring the mobile phase into the developing chamber. The one which you see is the developing chamber. And she is placing the the plate which has been applied with these samples, and she permits it to get eluted. After elution, the developed plate is seen now. The plate is looking like this. So we selected the colored compound for easy understanding. If it is not colored compound, then we have to go for the viewing to the UV chamber. So since it's a colored compound, it is visible, so we don't go for the UV chamber. From this, you can make out 3 components are separated. You just watch the right spot, which is numbered as a 5 at the bottom, and it is of S1 solution. So you can see three spots. The top one is because of the curcumin. Second is due to dimethoxy curcumin. Third one is due to the bis dimethoxy curcumin. And these solutions are lower concentration, which we prepared 0.01, 0.02, 0.03, 0.04%. Now I want everyone to watch with which solution, with which one this bis dimethoxy curcumin matches in size as well as in intensity. If you closely watch, you can find out it matches with the solution 2 and solution 3. That is, this bis dimethoxy, the intensity and the size of the spot due to the bis dimethoxy curcumin matches with the principal spot obtained with 0.02% and 0.03% solutions. So the component, the material which is present here would be same as that of this. So if you find out what it is, then we can say what it could be. From this, we can calculate what is the concentration of this bis dimethoxy curcumin in the total curcuminate complex. So I'll show that. See, this is the formula. Let's assume that the bis dimethoxy curcumin, let's say it is a X. As per this procedure, if size and intensity of spot of any component say X. Is it audible? Somebody is speaking. As per this procedure, if size and intensity of any spot of any component say X, obtained with the solution of higher concentration, in this case 1% solution, just watch the photo at the left side, match with that of the principal spots obtained with the lower concentrations which are applied on the plate. So in that case, the formula to be applied is percentage concentration of X in the sample is equal to LD divided by HC multiplied by 100. LD is the concentration of the solution, the principal spot of which matches with the X in the solution of higher concentration in terms of size and intensity. That is either 0.02 or 0.03. HC is concentration of the solution of higher concentration. That is, in this case, it's 1%. So applying this formula, if you assume that the BDMC is matching with 0.02, then 0.02 divided by 1 multiplied by 100, that is 2%, or it can be 3%. So since it is the size and intensity of the BDMC in the 1% solution matches with the 0.02 and 0.03, I can say that concentration of BDMC in the 1% solution is somewhere between 2% and 3%. Since we are not exactly saying what is the percentage, we call it as a semi-quantitative analysis. So for urgent analysis, we can adopt this mechanism where we don't require anything to find out whether we don't require even a UV spectrophotometer to detect this. Just a UV chamber is okay if it is a white sample, uncolored sample. Okay, I hope this is explained to the best of my capability. I'm going for the next slide. I'm skipping these 2 slides because this is explanation of the formula, what I arrived at in the previous steps. Because it's too complicated, I'm skipping these 2 slides. In the question and answer session, if anybody wants to explain this formula, I can come to this slide. Slide number 17 and 18 I'm skipping. Now I'm taking, with the completing the explanation on semi-quantitative analysis, I'm taking up real quantitative analysis by TLC. If you see, in this case, I'm taking finding out total curcuminoids present in turmeric rhizomes. This is what I'm taking as a case study. With this case study, we'll try to understand how we can conduct a quantitative analysis by TLC. Okay, we are taking. Okay, so I'm continuing. See, let us take first working standard. 10 mg of curcuminoids working standard is accurately weighed and diluted to 10 ml in a standard volumetric flask. That is, it is 1 mg per ml. Then I'm taking a sample, the dry turmeric. We are going to estimate what is the total curcumin present in that turmeric sample which we are taking now. So we are taking 25 gram of dry turmeric and it is getting powdered, and 2.5 gram of that powder is taken in a 100 ml beaker. 20 ml of acetone is added and it is stirred for 15 minutes. Then we are allowing it to stand for just for 5 to 10 minutes. So we are taking the clear portion of the acetone separately in another beaker, and this process of adding acetone, stirring and collecting back acetone is repeated for 3 to 6 times. And almost all the curcumin present in the turmeric powder would have come in the acetone, and it is called acetone decoction. This acetone decoction is again evaporated to the level. It is concentrated by heating to the 40 ml. Then again we dilute it to 50 ml in a standard volumetric flask. So the first solution is now ready for the testing. That is 2.5 gram in 50 ml. This is called S1T1. This sample, the solution ID is S1T1. The concentration of turmeric in that solution is 50 mg per ml. Then from that we are going for a serial dilution, which will have 50, 40, 30, 25, 10, 20 and 10 mg per ml of turmeric in it. And they are numbered like this, S1T1 to S6T6. And now we have applied all the solution, that 7 solutions, S1, T1 to S6, T6, with the working standard. 7 solutions are applied on a plate, 10 microliter accurately, quantitatively applied, and the plate is, the chromatogram is developed, and the developed chromatogram will be looking like this. And now we are scratching the spots obtained due to WS working standard. 3 spots because we are going to calculate total curcuminoids. We are scratching all the 3 spots and collecting a stopper test tube. Likewise, we are collecting the scratches of all the other sample applications. So we will have a 7 stopper test tubes. With every one, we will accurately pipette out 10 ml of acetone and shake for few seconds or few minutes. Then allow to settle down. And take the clear solution and measure the absorbance at 425 nanometer. If you need, you can go for the UV spectrum also. If it is unknown compound, if you want to know what it is, you can go for the UV spectrum also. From 175 nanometer to 750 nanometer, using your spectrophotometer, you can measure it. So now I've taken the absorbance, which is tabulated here against the concentration of each of the solutions which we have prepared. So you can see it from 50 mg per ml to 10 mg per ml solution concentration. We are having a 0.442 to 0.091 as a absorbance, and using this concentration and the absorbance what we got and comparing it with the concentration of working standard what we have taken and the absorbance what we have taken. Applying the formula given at the bottom, we have calculated what is the total curcumin present in the sample, which is 2.71, 2.71, 2.76, 2.70. The same sample with a different concentration of solutions, we have got almost similar results, the average of which is 2.74%. Interestingly, the same sample when it was analyzed with HPLC, we have got result as a 2.7% as total curcuminoids, which is almost matching with the average what we have got here. And the relative standard deviation here is 1.45%, which is a measure of reproducibility, I can say. It is not only a kind of an approximation, but we can take this as a reproducibility or repeatability, whatever the way you want to use. Then next slide is, and since we know the concentration of total curcuminoids present in the sample what we have taken, instead of the concentration of turmeric present in the solution, I'm applying 2.74% to this 50, and I'm calculating what is the concentration of curcumin in each of those solutions. So if you see S1T1 has 1.37 mg per ml and S626 has a 0.274 mg per ml as a concentration. And I'm taking the same absorption what we got the others shown in the other slide, and I have tabulated the concentration of curcumin in each of those solutions and the absorbance what we have got, and we have plotted a graph of concentration of curcumin against the absorbance what we obtained. It is looking like a straight line which is passing through the origin. So it means that this method is giving a linear results within the range of 0 mg per ml to 1.37 mg per ml. Now I'm going to the recovery test. I'm not going to explain what is the different solution, how we prepare and all. It is a bit tiresome. In the question and answer session, if anybody is particular, I can go for it. Now I'm confining only with explaining the results. In the recovery, we conducted 6 different recovery tests. We have got 101.42, 101.58, 98.8, 100.85, 100.71, and 99.38. It's excellent within the expected result. The error what we can see is 1.58%.
Dear participants, you can mute yourself.
One second. Is it audible now?
Yes, sir. Yes, sir. You can go ahead.
So in the recovery test, I'm not explaining how we prepare the solution. The net result only I'm going to see. Okay, who are they? Somebody has raised their hands, it seems. Can you please tell me what it is, Vignesh or Dr. Manveli?
I think Mr. Sarangan and Mr. Pinto has raised their hands. Could you please?
Can you please?
So this is Sarangan.
Tell me, Mr.
Sir,
Sarangan.
I had actually raised the hand for when you were explaining the thin layer chromatography experimentation pictures. So when you actually spot the sheet, doctor, so how do we ensure that all of the spots are exactly same quantity of solution?
We are using a micro syringe of 10 microliter.
Okay, and that accuracy of that system is fairly good.
Accurate. Everywhere we are, even in GC, we are using that.
Okay, sir. Yeah, that's what.
But it requires some expertise. In GC, we are using it, but to avoid the error, normally we use internal standard in GC, which is not possible in TLC, but still we need an accuracy here. In GC, the error will be very high if you don't inject properly, because injector port will be having a very high temperature, say 200 degrees centigrade, whereas this is a room temperature and the error will not be there. But with the experience, one can do it without any problem. I've done many times, and my laboratory is using this principle.
Okay. Yeah.
Any other question? Shall we proceed? See, I hope we can proceed. Is it Vignesh? Sorry?
Dr. Pin, do you have any questions?
No, let's not take any question now. We'll proceed.
Okay.
Unless it is very difficult to go further for them, they can stop us. Otherwise, the flow will be disturbed for others. So
Okay,
now I'm resuming the recovery test. So I'm not going to explain the protocol adopted by us for the recovery test. I'm just going to explain how much result we have got as a the recovery. So it is from 101.4 to 101.58. The maximum error what we have got is 1.58. That is, the method is the accurate enough from the level of 98.5% to or 98.4% to 101.6%. Okay, yeah, 100% sample can give either as a 98.4 or 101.6. So this is the error that we can expect in this method. So I'm going to the next slide. Slide is not moving. So with these things, I'm trying to consolidate what are the things we have seen in the quantitative analysis. Number one is the linearity and range. So we understood that the method is very linear from 0 to 1.37% mg per ml concentration, and it is accurate enough between 98.4 to 101.6, and it is precise to the level of 1.45%. That is, it is reproducible to the level of 1.45%. Specificity, the method is very specific because the spots what we obtain with the sample is exactly matching with the samples what we obtain with the working standard. So method is very specific. And the limit of quantitation and deduction, we did not do any test specifically to find out what is the exact deduction limit or what is the quantification limit, quantitation limit. We restrict ourselves only with the samples what we carried out, and it is 0.27 mg per ml is the deduction as well as quantitation limit. This we are concluding for this session. And ruggedness, robustness is nothing but carrying out the similar analysis by different people. If you get the same results when we carry out to the different people, and if you get the same results when we carry out at a different laboratory on different times, if you do all those things and still if you get the same results, then this method can be declared as a rugged and robust. And since we are following this method in our laboratory for the years together, and we get always acceptable results, we conclude that it is a rugged and robust, and we did not take time to explain with a different set of experiments that it is rugged or robust. So I'm not taking it further. It will take a lot of time, so I'm stopping this. With these things, I am confident, and I'm very sure the quantitative analysis by TLC is no way inferior to HPLC. And let us really see, we'll compare in the table on the next slides, then we'll try to understand still more in a better form, in better way. So let's see, just to see the tabular column here, the stationary phase in each of this method in the TLC as well as HPLC, in both the cases, it is a silica gel. The mobile phases in both the cases same, mixture of solvents. It can be organic solvent or triethylamine or buffer or acetic acid, something like that. So the mobile phase is also same. And the detection means in both the cases we are using UV spectrophotometer, UV light. Basis of qualitative identification is response factor in TLC, and it is relative attention time in the HPLC. Both are corresponding with each other. Both can be matched. So in principle, both are working under the same principle. So there is no way we can say that this is different from that. Both are one and the same. Only thing is HPLC is working with the support of pump and an automatic spectrophotometer, which is absent in the TLC, which we are doing with the manual means. Okay, now next question is number of samples that can be run in a given time. We can run any number of samples on a single plate if the sample is same. That is, if it is turmeric, And if you have a 5 different batches, 5 different lots, and all those samples can be applied on a single plate, and we can have only one working standard spotted adjusted to them, and we can analyze and we can declare the results after developing the plate, which is not at all possible in HPLC. And can we have a UV spectrum of the components? Yes, it is really possible, that too very accurately in the TLC, which is not at all possible with normal regular model of HPLC. Okay, I mentioned here normal regular model of HPLC, which I will explain. I will come little after explaining this TLC. In TLC, after scratching the spots, collecting in test tubes, measuring, we can measure in a cuvette the absorbance. And when we are measuring the absorbance, we can even opt to go for the recording the chromatogram spectrum from 175 nanometer to 750 nanometer, and using which we can make out what kind of component it is. Qualitatively also we can make out. Whereas it is not possible in a normal isocratic system which is fitted within a normal UV detector, because it is only one wavelength can be fixed in the HPLC, and the pump, you cannot measure. Say, for example, if the components are absorbing a different wavelengths, say one component A might be absorbing at 270 nanometer, component B might absorb at 295 nanometer, third one will be absorbing at 350 nanometer. So in such case, you have to have 3 runs measured at 3 different wavelengths, unless you have a photodiode array detector fitted with your HPLC. The fitting of photodiode array detection will be very expensive. So can you please mute your hello? Can you please mute? Yeah. So shall I mute everyone here?
Yes, I think.
Yeah, I muted. So now I'm going back to my screen again. Okay. See, as I explained you, in TLC we can have in a single run we can see the UV spectrum, and even if those components are having a different lambda max, we can measure. Whereas in HPLC, in normal HPLC, normal isocratic system fitted with a normal UV detector, it's too difficult to exactly get the peak area. Because if component A absorbs at 254 nanometer, and if you fix your wavelength of detection is 260, this 254 nanometer component will give us smaller peak. So it's a very complicated. I don't want to explain it. I'm leaving this to imagination of the participants here, because almost everyone is familiar with this system, I believe. If it is not familiar, you ask me once again during the question and answer session. This is my interesting area. And can we run 10 different samples at a time? Yes, we can run 10 different samples. In the point number 5, I explained same sample, any number of samples. Here, we can run different samples with a different developing chamber with a different mobile phase at any given point of time. And we can get the results of everything together. Whereas in HPLC, it is not at all possible. Every time you have to inject a separate sample. And the time required for the analysis, in TLC we require only 10 minutes, for maximum 10 minutes for completing a phone analysis, that is for development alone. Then scratching and measuring by UV spectrum may take additionally another half an hour. But in HPLC, as per the method recommended by USP, System Suitability Procedure, you have to inject minimum 6 times. The working standard injects 6 times, or the sample might be 3 times, I believe. I don't remember now. So in any case, a single analysis might require 3-4 injections, and each injection might take 20 minutes to 60 minutes. So ideally speaking, you cannot analyze more than 2 samples per day if you're going to inject multiple times in your HPLC column, whereas it is very much possible in the TLC. Then the cost of the equipment, if you see, with the TLC, it is just 1,000 rupees. Whereas HPLC, if anybody is giving with a discounted price, it can be 8 lakhs for an isocratic system with a normal detector, UV detector. If you don't get any discounts, it may vary from 14 lakhs to 25 lakhs to 30 lakhs. Okay, runs into lakhs. And the running cost, if you ask me, the analytical cost it costs only 10 rupees for the TLC. 10 ml of solvent, 10 rupees. Whereas in HPLC for solvent cost alone 75 rupees. And since it is having a lot of moving components, the wear and tear is very high and the depreciation rate is very high. And the maintenance of a HPLC when you run it on day-to-day basis, it is something like having an elephant at home. Okay, so let me explain you what is the maintenance cost involved. In TLC, it's nothing. In HPLC, yeah, annual maintenance contract, I don't think it will be less than 25,000 nowadays. It's quite a while that I didn't engage anybody so far in the recent days, so I don't know what is exactly the annual maintenance contract fee. Then lamp cost, if you want to have an ideal system, the lamp has to be changed for every 500 hours. The lamp cost will be, I just judge it will be 20,000, and column is easily 20,000 rupees. And the column is to be changed for every 900 injections. And the plunger seal cost is mostly, this is a rough cost, which I don't have the accurate cost. It will be around 12,000 rupees for 2 numbers because it is having a reciprocating pumps. And as per my understanding, it is to be changed for every 1,000 samples. So with these things, I leave the choice of taking decision, which one is better, whether it's a TLC or HPLC. Okay, so for my choices, I opt for TLC, and we are running a very good laboratory. All our developmental work is done only with the TLC, both quantitative as well as qualitative. See, we have developed a process to develop extract lutein. And we have developed a process to extract rosemary, the carnosic acid from rosemary. And we have developed a process to extract zeaxanthin, or to convert lutein into zeaxanthin. All these processes were followed up with the TLC. And as a policy, we don't have HPLC in our laboratory. And all our tests, all our assays are calculated using TLC. And we are exporting currently to different countries like Australia, New Zealand, Germany, Italy, France, and Canada. And all these countries, we are sending our material without analyzing using HPLC and with analyzing using TLC alone. So I'm leaving the choice to the participants to decide which one is better. And I don't say that HPLC is inferior. HPLC is equally good or slightly better also in some cases. Where it is really required, we cannot avoid using HPLC, but where it is not required, why should we use TLC? In my opinion, we are using HPLC where it is not really required to be used. With this, I'm concluding. I take this opportunity to thank Ms. Priyanka, who has carried out this work as a part of the project work, and she was an MSc Chemistry student from Katpagam Academy of Higher Education, which is popularly known in our area as Katpagam University at Coimbatore. She stayed with us for around 3 months, and she completed this complete analysis, and she produced a very beautiful dissertation, and she was guided by our R&D executive, Ms. Siva. And I thank both of them for completing the study, and I take the liberty to explain this using their work. Thank you, Siva and Priyanka. Now I'm leaving this session to the question and answer session. And my name, my number, my email ID is given here. I just leave it for some 30 seconds. Anybody wants, you can make a note of it. And now we will go for the question and answer session. Okay, we'll take up the question and answer session.
Sir, good evening, sir.
Good evening.
I'm Dr. J. Kumari, working in the phytochemical analysis. A very good presentation, excellent presentation, sir. Sir, when you are doing the for herbal extract, sometimes it find very difficult, sir, to compare with the standard. If a standard is available, we can go for quantitative. In what we'll do, sir?
This is, we have to somehow manage to get this, the working standard, whether the authenticated standard you have to get. We normally get authenticated sample and use that as a working standard, and we compare. It's a qualitative. As long as it is qualitative, we have to go for authenticated sample. Nowadays, a lot of people are offering it. Even the natural remedies private limited, NRPL is offering authenticated sample, I believe. To the best of my knowledge, we prepare ourselves and we do a lot of characterization study. And at a later stage, we'll use it as a working standard. We develop working standard with respect to the authenticated sample, and we compare it with the working standard.
Thank you, sir. Thank you.
Thank you. Thank you. There is one question from Venkatesh K. What about the accuracy of TLC compared with the HPLC? If it is, so I showed you in the presentation, it's almost same. If it is properly done, it is almost same. In the HPLC, we got 2.7% as a total curcuminoids. Same sample when it's analyzed using TLC, it is 2.74%.
So sir, I have one doubt actually.
Yes, tell me, Dr. Tangamani.
I think if you talk about the TLC and HPLC, I think for the, if you do for some import for the material, if we say that the HPLC and the TLC, we have the same accuracy.
Yeah.
Yeah. What about your opinion about that? People will, we are doing the TLC, we know that we are done in our laboratory only.
Yes.
We do not get any reports actually. We have only photographs.
Yes.
But HPLC, we generate the report, whereas our user will asking that the purity, we will send the report, right?
Yes.
So what about that, sir?
So do they believe that peak report what you sent?
Yes.
Okay. And how do you establish that? What is the concentration you injected there?
Yes, sir.
Now, how do you establish? How do you make them to understand or accept? What is the record that you have? What is this, how you prepare the sample solution for the injection, and what is the concentration you prepare? You say that it is a 10 microgram per ml.
Yes, sir.
But I say that you are not taken 10 microgram per ml. You have taken 12 microgram per ml, and you have got a better peak height. And
yes,
so if I say that, how you can deny it?
Yes, sir.
So only because you are having a chromatogram on the paper, you cannot say that it is a proof. So when you cannot say that it is a proof, TLC also does not have a similar proof. HPLC, I don't think it is having a, since you are having a printout in your hand, you cannot say that it is a proof.
So somebody will validate actually. For with that validation, any proof is not acceptable, sir.
Sir, actually validation is in TLC also we are validating. I showed you how we are validating.
Yes, sir.
In HPLC also it is validated. But
Yes,
now you are saying that you are making a product. That product is analyzed by HPLC, and you have got so much of peak area by showing the peak report, and you expect them to accept this. But my question is, they will accept the peak area, but what is the concentration that you used? Do you have any records for it? You could have injected 12 mg per ml, and and you could have you could have calculated by saying that it's only 10 mg per ml, so that the purity is high.
Yes, sir.
So absolutely there is no proof for anything.
Yes, sir. Yes, sir.
It's only a kind of a psychological feeling that when you have a chromatogram in your hand, it's everything is perfect. Correct?
Yes, sir.
Sir, in continuation with this question, I also have a doubt. So what I understand from your answer for this particular question is, me doing research, when I submit the TLC reports, they say HPLC has the better, precise, accurate results. And they ask me to give HPLC result, and they don't agree with TLC. So from your answer, how I understand is, it's a way how we explain and establish with whatever papers we have in our hand. So you have the luxury of saying, you can just say how HPLC is so accurate, but as a research student, I cannot go and argue with that point, no, sir. So can you help me how to establish with the result which we have with TLC?
Yes. This is a typical situation which I came across, and that is the cost for preparing such a kind of an extensive tabular presentation a while ago. Actually, we applied our laboratory to get recognized under DSIR as an industrial research laboratory, and we were denied that status by the DST, Department of Science and Technology, saying that you don't have the HPLC. Then I asked them, the panel members, the questions, and I explained them HPLC is no way better than the TLC, and till the end they did not accept. So I didn't pursue it further. I left as such, and it is difficult if somebody is not interested in getting up, or it's not listening to you. We cannot do anything. But generally speaking, the USP accepts it. See, in our industries and all, it is commonly accepted. It's a common. Many people do not accept. Many people do not come out with the feeling of saying that HPLC is far better than the TLC. See, before HPLC becomes very popular, that is, which happened only after 1995. Till then, only the very few number of HPLCs there in even in entire Chennai. Okay. Even before that, how this quantification were done. I explained you in 1985 in Indian Pharmacopoeia. It has been accepted, part of the TLC is accepted as a quantitative means. So when an official organ is accepting one particular method is official, how can it become, or it's an authenticated one, how it can become not acceptable when the advanced system becomes popular? What I feel is, in our industry, if you read the USP or IP, I don't know whether you are aware of USP and IP. And the IP is Indian Pharmacopoeia, which is a standard prescribed by the Indian government for approving every drug. And the USP is United States Pharmacopoeia, which has official methods. USP, if you see clearly, read through, they have clearly mentioned the methods given here are only for, it's a recommended to be used in your laboratory. If you have a better methods or equivalent methods, and if you can validate at any point of time, you can have the liberty of using your own method. So using that statement, we are doing a lot of tests on our own. And if anybody questions, I am very confident that it can be compared to the HPLC. So you can talk to your reviewers and you can tell them this method is validated, and you can give the validated procedures to them. Mostly they will accept. I'm not very sure about it, but in industry and the regulatory departments, they accept.
Thank you, sir. That's a valuable input for me because I can refer USP and IP and I can establish my point there. That's a very good point. I got it from you, sir. Thank you so much.
Thank you.
Sir, we have a question from Ms. Lavanya. You have
Yes.
told that TLC has got a lot of advantages, but in which cases HPLC becomes essential?
It's a very difficult for me to answer. See, sometimes the application, see the accuracy, if you see, it is between 1.6%, plus or minus 1.6%. In HPLC, I have seen up to 0.5% in certain cases, but it all depends on the people who use the technology. See, in my opinion, we are very comfortable with the TLC, and we never go for the HPLC so far. And yes, it is very difficult for me to reply to this question, which requires or which doesn't require. For me, so far, such a requirement never came up. But nevertheless, we cannot ignore totally HPLC.
Thank you, sir.
Thank you. Yes, sir.
Good evening, sir. This is Dr. Kannan.
Yeah.
Chiral compounds can be separated and quantitatively analyzed by chiral column in HPLC. Can we do that in TLC also?
It's very difficult. Actually, the chiral stationary phase is something different from the silica gel what we use. Silica, that's one reason we can take the Lavanius, this answers Lavanius question. We do not have such a kind of stationary phase so far in the TLC, but to an extent we are using magnesia to separate the isomers. I really do not know whether chiral compounds can be separated in the magnesia. I have not experienced. But what we do normally is we will make the component, if it is in a basic material, we will make them to react with some tartaric acid or something like that. And we'll convert them into diastereoisomer, then get it spotted. So that we'll get a different spots.
Thank you.
Thank you. Is it clear? Does it answer your question?
Yeah, understood. But in HPLC, even the stereoisomer can be separated. We don't have to convert it into diastereomer.
Yes, because the stationary, they have some liberty to use a different stationary phase, which is not, such a liberty is not there in TLC. Unless you convert into the diastereoisomer, we cannot do it.
Yeah, okay. Maybe this may be advantages of
one of the limitation of TLC.
Thank you, sir. Sir, one more question in the chat box, Dr. Subhashini. In your purview, what is the scope of phytochemistry in the present era?
I think that's very bright. Actually, we want to make this possible, and that is the reason why we have initiated this Geern Pharmaceutical Initiative. This is a different topic as such, but anyway, I'll take just 2 minutes to answer this question. She is a very senior person in this industry and academic. And my opinion is almost every drug is having one or other phytomolecule as a model. If you take vincristine sulfate and vinblastine sulfate, they are synthetic analogs of the extract got obtained from the Vincorossia. And if you take dextromethorphan, it is a synthetic analog of morphine or codeine, which again are phytomolecules. So if you see the different phytomolecules that can be extracted out of the herbs, mostly we can take Siddha as one of the input. If you take the various herbs explained in the Siddha and start extracting every herb, we'll get a series of phytomolecules, and all of them will have a medicinal value. And if you document them for the safety and efficacy, probably we can slowly move towards a greener system than the current synthetic system. Currently, the atom economy of synthetic, when we synthesize drugs, the atom economy is only 37%. 63%, it means that when you make the 37 gram of a drug, 63 gram of the other chemicals are going as a pollutant to the environment. So in order to avoid that, we have to go for the phytopharmaceuticals. And I wish it has to happen very shortly, but I don't know how long it will take. We have started building the cat. Let's see.
Okay,
So thank you very much. After a long time, I could join your webinars, and I'm happy that I did not miss it. This is Subhashini speaking. I could join a little late only, but I found that your emphasis on taking forward the work with minimal of expense is indeed a necessity for generations of workers in India, especially in this field. Maybe the final validation we can go for HPLC or HPTLC, but as you rightly said in the pharmacopoeia, what was initially involved was the primary methods, including TLC and the fingerprints, TLC fingerprints and some NMR if it is there. They have mentioned in the pharmacopoeia for each of these drugs. So phytochemistry, in my view, is a poor man's research, but still we can definitely do lot of work, quality work with very minimal expenses. The only effort is the dedication and the hard work is the expense, is probably the whatever should be the expense from the persons. So thank you very much, sir, and you're doing such great efforts to educate especially the younger generation. Wish you
Thank you,
all the
madam.
best and wish you all success, sir.
Thank you. Thank you, madam.
Thank you. Thank you very much.
Thank you, ma'am. You have couple of questions in the chat box. I'll read out one after the other. This is a question from Dr. Ponmari P. Whether biological samples can be separated by TLC?
Biological sample means what are they?
I think it's like your blood, urine samples.
Yeah, any component, any chemical compound. If it is a chemical compound, they can be separated. We have used the blood serum as a sample to find out the presence of dichlorophenoxsodium after consumption. After every 1 hour, they used to take blood and prepare serum out of it, and they will give to us. And we have used TLC and we have estimated the quantity of dichlorophenoxsodium present in the blood serum. Also in the mother's milk, we have taken, found out whether it has anything. Instead of injecting in the HPLC, we have done using TLC. More accurately, we have done it. It's possible.
Okay, thank you, thank you. Next question is from Dr. Santosh R. Is there any other different study conducted that you prove TLC is more efficient than HPLC?
I really do not know. This is my own analysis. I never referred to anything, and I'm in this industry for a quite long time. Say around I started my career in 1987, and those days, I can say that probably I would have been the first person in South India who used a PDA detector in HPLC, and I compared HPLC analysis many times with the TLC the last 30-35 years, and I'm pretty sure that certain things which cannot be detected in the HPLC we have detected in the TLC, and because of which my career has grown to a greater level. So I can cite out one example is when I was working for a company which is known as the UB United Breweries Pharma Division, we were making ranitidine. So in ranitidine, one impurity, one related substances should not be there beyond a level of 0.05%. In HPLC, it did not find out, whereas in TLC we could find it out, and we proved that one of the well-known company in India, which was making that time, has the impurity beyond 0.05%, which we proved, and we made them to understand, and it helped me to get an entry into that company as an head of the QC department. And I don't think there is any study as such, but someone has to initiate. This is out of experience I'm talking this.
Next question in the chat box by Mr. Syed Mohammed is it possible for all compound to identify by this method?
Whatever you can do with the HPLC, you can do with the TLC. That much I can say. It's not that everything can be done, but it's limitation-wise, both have similar limitation to an extent. But almost all the samples, except the solvent samples, anything which can get absorbed can be detected.
Even Venkatraman sir, Dr. Venkatraman sir from USA has replied to Ponmari question. Whether biological samples can be separated by TLC, many lipids can be separated using TLC. He has replied in chat box. Thank you, sir. Thank you. She has also replied us. Thank you, sir. Thank you. Thank you so much for helping us in the online platform. Any more questions? And Dr. Pintu Kumar, you have raised your hand. Can we have on the board? Can we have you on the board?
Yes, I can.
Dr.
I can view.
Pintu, do you
Mr.
have
Pintu
any
can
questions?
answer. Can question me now. I think he has just raised. Can you read through the chat box? I don't have access to it.
Yes, not type these questions. Yes, just raise his hand.
He has said that there are no more questions. I think there is a message from Mr. Pintu Kumar.
Okay, so I'm also seeing it.
Any more questions? Participants? Any more questions?
I got a call from that. No other question means we can. I think there are no questions in the chat box as such. Can we share the feedback form link so that?
I've shared.
Yes.
Participants, you can fill up the feedback form.
Okay. Thank you, all the participants, all the dignitaries, whoever I respect very much. And all the participants, student participants, I thank all the people who take time to make it convenient, who made it convenient to attend this program. Thank you. Thank you for everyone. Thank you to everyone. And if there is any question, just give me in your chat box, or you can talk through the audio system mic.